All right, so we can probably go ahead and get started now. So hi, everybody. Thanks so much for coming to our CCN community event. So you have a highly predictive encoding model. Now what? Translating neural encoding models into scientific and engineering impact. My name is Maggie Henderson. I'm an assistant professor of psychology and neuroscience at Carnegie Mellon University, and I co-organized this event along with Janelle Feather and Jacob Prince. We're very excited to share with you today a really great series of talks and a panel all on the big topic of how we can use predictive neural encoding models for scientific discovery as well as for engineering and translational applications. And before we dive into that, we want to start with a little bit of background. So we're going to lay out some of the basic ideas behind encoding models and how we use these models in cognitive neuroscience. So the general setup with these models typically is that we have some neural data that we record from a participant in response to a stimulus. And this can be broadly applied across many domains. So this could be visual stimuli like images and videos, can be auditory stimuli, can be language stimuli or others. We might then be recording from the brain of a human or an animal participant using methods like fMRI, EEG, MEG, non-invasive recordings. But in all these contexts, the goal is going to be to build a stimulus computable computational model that we can use to map between input stimulus and the resulting brain response. And typically we do this in a series of steps. So first we're going to extract a set of features from the stimulus using a computational model. This can be a deep neural network, for example. We can then learn a linear mapping from those extracted features to the brain signal of interest. For example, this might be an fMRI voxel. This might be a single neuron spike rate. We fit this linear mapping usually using regularized methods like ridge regression, for example. And this then provides us with a full predictive model that can map between any arbitrary stimulus and use that to predict the voxel or the neuron's response. And the final key step, of course, is to validate this model. The gold standard for this is going to be to take some held-out validation data that was not used during fitting of the model and see if the model can predict responses to that held-out stimulus. So we compute a measure like R-squared, maybe Pearson correlation, to capture the quality of prediction. So high R-squared means we have a highly accurate predictive model. And of course, the challenge you might face with this is that we'd like to be able to predict the response of the brain to arbitrary, naturalistic, complex stimuli. So this is where deep neural networks, or DNNs, have become really useful. So we can use DNNs to extract rich sets of features from stimuli, and we can then incorporate those features into this naturalistic neural encoding model framework and build predictive models. And since around 2014, as many of us know, there have been many landmark papers showing that the features we extract from DNNs can be used to predict responses to naturalistic inputs across a range of brain systems, from visual cortex, auditory cortex, the language processing network, somatosensory areas, and others. And these models can yield accurate predictions of responses. They've also been shown to have some degree of hierarchical correspondence at the level of model layers and hierarchically organized brain areas. So this all suggests there's some level of emergent representational alignment between DNNs and biological brains in terms of the representations and the computations they may learn, although the nature and the extent of that convergence is still very much debated. And so today, as a field, we find ourselves in a very exciting moment where we have continually larger and larger models. So this includes new machine learning model architectures like the transformer. We have models trained on bigger data sets, internet scale data, multimodal training schemes that incorporate images alongside text and videos. In neuroscience, we're keeping up with that progress by collecting new large neuroscience data sets or neural data sets, including big fMRI efforts, the natural scenes data set, the neuromod data sets, similar efforts in systems neuroscience. And to track that progress, we've begun to develop platforms for model benchmarking in a standardized way, including the BrainScore platform, the Algonauts competitions. But so given that we have these very big and very predictive models, and they continue to improve, what kinds of things can we then do with these models? And so this is going to be the focus of today's event. So we're going to start by giving a little bit of a quick landscape of what kinds of things we can do with these models. So broadly speaking, encoding models can be useful on both the basic science or theoretical side and on the translational engineering side. So on the basic science side, probably the most popular way to use encoding models for science has been the model comparison framework. This also includes things like variance partitioning. In this kind of a scheme, you might take multiple encoding models. They might have different constraints, such as being based on deep neural networks that have different architectures or different inductive biases. Maybe the models are trained on different data sets or different tasks. We might then compare the predictive accuracy of these models. And if you do that comparison in a very careful and very controlled way, we can use this to test hypotheses about the kinds of constraints that might be needed in order to learn a good model of a brain area of interest. But beyond looking at the predictive accuracy of models, other work has begun to study the properties of the encoding models themselves. And this has often been recently termed the digital twin framework. So the idea being that once we have a good predictive encoding model, we can then use that as a test bed for doing kind of in silico experiments. And this can include, for example, running large-scale, high-throughput tests that would be difficult to do in a typical biology experiment. On the more engineering side, recent work has also begun to incorporate encoding models into a closed-loop paradigm, where you can iteratively optimize a stimulus, sometimes in real time, to elicit a target neural response. And this can also be useful for basic science questions, such as identifying what's the best stimulus to drive a neural population, even characterizing tuning invariances. And there's also many promising applications of encoding models for translational purposes. So just to name a few of these, encoding models can be useful for improving the accuracy of prosthetic sensory systems by learning a precise mapping between a sensory input and a neural response. They can also be a component of improvements to brain-computer interfaces, or BCI technology. So in this case, an encoding model might be leveraged as the basis for a decoding algorithm, which decodes or reads out intended speech or intended motor commands from neural activity. And we're going to hear much more about these and other applications of models from several of our speakers later on today. But so with this in mind, we have these highly predictive, powerful encoding models, and they have the potential for big scientific impact across all these different domains. But it's clear that many open questions remain. We might ask, for example, what outcome measures are most useful for encoding models? Can prediction scores on natural images be misleading? Does modeling natural images actually tell us about the underlying neural tuning functions that we want to measure? Are there any other modeling pitfalls we should be aware of? What tools are best for probing and interpreting encoding models? Should we use stimulus synthesis? Should we develop other interpretability techniques? And how can encoding models ultimately be used to guide new experiments and new applications? And so this broadly motivates the topic of today's event. So we have a great set of speakers who are each going to be offering their expert perspectives and sharing how they approach these questions in their own research. And we're going to hear about some new avenues for leveraging highly predictive models to obtain new scientific insight and drive translational innovation. So our speakers for today, we're going to start off in the language network with Greta Takute from Harvard University, who's going to talk about predicting, controlling, and interpreting language responses in the human brain. Then we're going to have Nick Lessica from University College London, optimal hearing aid design through closed-loop neural restoration. We'll have Alex Williams from NYU talking about mechanistic interpretability of neurally predictive models. Then going over to the vision side, we'll have Iris Groen from the University of Amsterdam talking about leveraging encoding models for scientific insight in human scene perception. We'll have Thomas Nasaleris from University of Minnesota, concept dose responses in human visual cortex. Then finally, we'll have Johannes Mehrer from EPFL toward model-guided visual prostheses and model-optimized dyslexia-friendly fonts. And after that, we're going to have an interactive panel discussion with all these speakers, moderated by Janelle. And this is going to be a discussion organized around the key goal of identifying a set of concrete steps that can move our subfield forward towards more productive modeling efforts. We're going to ask what kinds of infrastructure, benchmarks, and cultural norms should our field be adopting in order to most effectively translate the data and the models that we have into new forms of scientific progress. And with that, I'm going to turn it over now to our first speaker for today. So we're going to kick things off with Greta Takute. Thanks, everyone. Take it away. Great.
Thank you, Maggie, Janelle, Jacob, for putting all of this together. All right. So as you listen to me right now, there's a set of frontal and temporal regions in your left hemisphere, the language network, that is supporting your ability to understand these words. And thanks largely to large language models, we now, as of a couple of years ago, have encoding models that predict responses in this network remarkably well. So now what? In this talk, I'll focus on two scientific questions where encoding models enable scientific insights that I would argue would have been impossible or very hard to obtain otherwise. Okay, so the first scientific question is, what kinds of linguistic input maximally engage these language regions? Basically, what is this preferred stimulus? A classic strategy in neuroscience is to ask what stimulus drives a neuron or a brain region most strongly. For instance, Hubel and Wiesel did this in the domain of vision by systematically varying the stimulus orientation, demonstrating how some neurons are most responsive to a particular orientation. And here we want to apply the same logic to language. However, one problem is that there's no straightforward sentence orientations to sweep over. The space of possible language stimuli is effectively infinite. So how do we know which stimuli to show? And I'll argue that we can use encoding models to search the space very efficiently. To do so, we first collected responses from 5 participants who each read 1,000 diverse sentences, and we fit an encoding model that predicts the response of each participant's language network to any given sentence. And this plot shows the measured brain response versus a cross-validated model prediction. And now we want to use our encoding models to basically identify sentences that are predicted to maximally drive this network or to suppress it, to elicit minimal activity. And to find these extremes, we used the encoding model to basically search over a couple of million sentences, or also a gradient-based approach to generate some new sentences. And then we selected 250 drive sentences and 250 suppressed sentences for experimental validation. So we tested these new sentences in completely novel participants. And this plot basically shows that the encoding model-based drive and suppress sentences elicited high and low activity in the language network, as predicted, basically demonstrating successful modulation of brain responses to language in a closed-loop manner. And I want to emphasize that this effect is actually not ubiquitous across the brain. This plot shows another set of brain regions involved in high-level cognition, the default mode network, that clearly does not show the same effect. Back to the scientific question I started with. Critically, because these sentences span such a broad response range, we could ask what kinds of sentences elicit maximal responses. To interpret these, I'm here showing the brain responses to all sentences in the experiment. Every dot is a sentence. And this is versus, on the x-axis, a crowdsourced participants' rating of sentence well-formedness, a kind of processing difficulty metric. And we have the completely well-formed sentences over here and the ill-formed sentences over here. And what we discovered was that sentences with a certain degree of unusual grammar and/or meaning actually elicit the highest activity in the language network. For instance, here, I'm progressive and you fall right. These are familiar English words that are combined in an unexpected way. So in other words, the language network is most responsive to the input that adheres to the statistics of English, but yet is hard to process. So regions in your brain that are really working hard to make sense of words and how they go together. And these results have opened up for a whole new alley of experimental and modeling efforts, and would not have been possible without an encoding model. Okay. So the previous result that I just talked about basically shows that the language network as a whole responds strongly to a type of sentences, those that are hard to process. But of course, that's only one aspect of language processing. So more broadly, can we distill the features that modulate voxel responses in this human language network? And this is a central question in language neuroscience, and it's a hard one. And maybe the rich feature spaces of LLMs can help here. We already know that these LLM-based encoding models are predictive of responses in these regions. So perhaps we can just inspect the features of these LLMs that account for each region's or each voxel's response. But it's pretty tricky because standard encoding models basically use many thousands of entangled, uninterpretable features. So we kind of run into the good old problem of relating one black box to another. And to try to get beyond this, we basically make two key changes to the encoding framework. First, to make the LLM feature space less opaque, we borrow from mechanistic interpretability. We replace standard LLM features with more interpretable sparse autoencoder features. And these autoencoder features basically project LLM features into a large dictionary space of features that are more selective and hopefully also more interpretable. And in our case, we use a 32,000 large dictionary space. The second change that we make to this encoding framework is that we explicitly include an estimate of a sentence processing difficulty using surprisal as a proxy. That's a sentence predictability. Because we already know that processing difficulty drives responses in this network, as I showed you earlier. So, we should build on previous work and include these predictors that we know matter, such that we can cleanly disentangle the contributions from difficulty versus these SAE features. And this then forms our encoding model basis. So for each voxel, we use an L1 regularized encoding model. So the model can choose from 32,000 features, but is forced to only use a small subset together with surprisal. We used 7 CFMRI data from participants listening to sentences. And I want to emphasize 2 findings. First, our encoding model lets us tease brain regions apart. And here I'll focus on, just as an example, 2 regions. And these 2 regions, the middle frontal gyrus and anterior temporal language regions, have previously been shown to respond similarly to various linguistic conditions. And here I'm showing the predictivity in these 2 regions from the SAE features in turquoise and the standard LLM features in red. And first, almost as a sanity check, right, we can actually see that the SAE features do preserve brain predictivity. So we don't lose much here. And now, critically, we can compare these 2 regions. And I want to ask, how much does surprisal explain on its own versus the full model with the additional SAE features? And in the middle frontal gyrus here, we can actually see that processing difficulty, this surprisal feature, alone predicts the response almost as well as these SAE features, even though this encoding model can draw from a set of 32,000 massive features, right? So this is a lot. While conversely, over at the anterior temporal region, there's a substantial improvement from these SAE features, meaning that the responses in this anterior region basically are much better explained by these rich feature spaces that are obtained from large language models. Okay. And now, critically, we can actually zoom into the region where we know that SAE features actually explain more variance, right? And then we can actually inspect what are the SAE features that explain these responses. I'll illustrate this using an anterior temporal voxel. And in this voxel, it can actually be predicted from just 7 of these SAE features, which account for around 90% of its explainable variance. Meaning that LLM brain alignment can actually nicely be decomposed into these small, sparse feature sets. And critically, we can then actually inspect these exact features, and the mechanistic interpretability community has actually created tools for basically looking up for every one of these SAE features, what does it fire for? Which context really activated strongly? So by doing so, we can get a candidate tuning profile. For instance, we can see that feature number 40 corresponds to something about emotions and relationships that positively drives this voxel. Another feature here corresponds to abstract content, such as consequences. And we have an example of also a feature that drives this voxel negatively, which is about concrete content, such as scenery. And this basically provides a pretty distilled description of which features of linguistic meaning representations are present in this voxel or in these regions. And nearby voxels showed similar profiles, suggesting that there's local substructure in this network. And I want to emphasize, of course, that these are just hypotheses. They're not final interpretations. The features are highly predictable and stimulus computable, and therefore this method can provide the groundwork for really interpreting how these vary across regions and across time. So basically, by incorporating mechanistic interpretability into the standard encoding framework, we can better understand the substructure in the language network. And here I'm basically just summarizing how, in the first question, we can use encoding models to generate stimuli for scientific insights and new experiments. And for the second question, how we can use mechanistic interpretability to let us distill these weird opaque LLM spaces into more interpretable sets. And just as a final note, I'll obviously argue that LLM-based encoding models are very useful. But they're really weird. Like, they're really, really weird models of language, and it limits theory building and any inferences that we can make about, for instance, language acquisition and processing stages implicated in language. So thank you.
I think we have time for one question between speakers.
LLMs start from subword tokens that basically mess up basically anything that happens in the system afterwards. It's like an unusual representation. And of course, humans don't get subword tokens when we learn. That's one example. It's completely feed forward. Yeah, I guess those would be two weird examples. Thanks.
Great, let's thank our speaker again. Great, and next we have Nick Lesica from UCL telling us about optimal hearing aid design through closed-loop neural restoration.
Thank you. And thanks very much for the invitation. My group works on hearing loss, and a big focus for us at the moment is trying to improve the signal processing in hearing aids. Many of you might not have thought that much about hearing aids, but they're quite an important tool for helping people with hearing loss have some relief from the perceptual difficulties that they might otherwise have. And they work quite well, especially in a situation like this, where it's generally quiet and rather simple in terms of the auditory scene, but they don't work very well in other kinds of important social settings, restaurants, on the street, and things like that. This is a long-standing problem that people have been trying to solve for quite some time, but without much luck. And it's because the problem seems to be beyond the scope of what classical engineering approaches can provide. But as I'll tell you, we're now trying to take a more data-driven approach, taking advantage of neural encoding models and hoping to make more progress that way. It's a big team effort. I just want to give a special acknowledgement to Fotis Dracopoulos, who's the postdoc who's doing a lot of the heavy lifting on this work. So I'll just start with the kind of cartoonish version of how we think about this problem. This is my auditory system. So we've got some speech coming in, being processed by the ear and brain, and giving rise to a percept. In this case, it's the word bat. If we try to crack open the black box a little bit, we can think about the neural code that underlies this. And this is kind of the key perspective that we take. So there's some specific pattern of activity that's giving rise to that percept. If we think about hearing loss simplistically, we might think of it as a loss of audibility, meaning the percept just becomes a bit weaker. And if that was the case, it would have to be because the underlying neural activity also became weaker. But that's not really all there is to hearing loss. There's also a loss of intelligibility. So in this case, the word bat might be misperceived as the word dad. And if that's true, that can only be because the underlying neural activity has not only become weaker, but it has also become distorted in some way. So if we take current hearing aids and think of them basically as simple amplifiers, all they are doing is amplifying that distorted activity pattern, which of course then just gives rise to a strengthened but still distorted percept. They're basically restoring audibility only. What we really want from the ideal hearing aid is for it to transform sound in whatever way is necessary, such that the sound that comes out of the hearing aid when it's processed by the impaired auditory system creates the full normal neural activity patterns downstream. If that can be achieved, then we will have, in theory, restored both audibility and intelligibility. So as I said, this is not even necessarily a new way of thinking about the problem, but it's something that really classical engineering couldn't really be used to solve. So we can think about it differently. We can replace the current hearing aid with a neural network, and we can try to let it learn from data what it needs to do to sound in order to get those activity patterns back to normal. As long as we know the target neural activity patterns that we want to steer the activity in the impaired brain toward, then we can hopefully train a network to do that. And if we do, then what we'll have is this ideal hearing aid that we're looking for. So the way we do this is to take a kind of indirect and two-stage approach using a neural encoding model, because this isn't something that can really be done directly from data with human listeners. If you put an EEG cap on somebody and try to train a hearing aid in order to steer neural activity patterns back to normal, it's not going to work because EEG just simply don't have the resolution or the signal-to-noise ratio required to give you the rich representation of sound that really makes the difference between bat and dad, for example. So we use animal models. That is the kind of key to our approach, and I won't go into the details too much. I'll just say that what we do is make intracranial recordings from gerbils. Gerbils are a rodent with very good low-frequency hearing, so they're commonly used for the study of auditory processing. And we record from the inferior colliculus, which is in the midbrain, partway up through the auditory pathway. And it's a place where there's already been a lot of transformation beyond what happens in the ear, and it's kind of the last stage where everything comes back together before it gets fed into the thalamocortical loop. So we record lots and lots of data from gerbils while they're listening to a whole range of sounds. And we use those data to train encoding models. We call these models IC nets because we're modeling the IC. And these models are trained to reproduce the data that we record experimentally as faithfully as possible. Once we have those encoding models, then we can train the neural network hearing aid. We call that AidNet. And in this case, once we've got the encoding models, then of course we're not data limited anymore. So we can use encoding models for normal hearing and encoding models for impaired hearing and put them into this closed loop framework, and that will allow us to then train the hearing aid with whatever sounds we need to. So I'll first show you a little bit about ICNet. It is very good at what it does. So it takes in audio, it puts it through, just as was pointed out before, a complex deep encoder to create some sort of latent representation that can be then read out through a simple linear layer to give us these heat maps of neural activity. So we record lots and lots of units with high temporal resolution. So these heat maps, it's unit on the y-axis, these are multi-units, and 1 millisecond time bins on the x-axis. For auditory information, you really need to use small time bins. So we get these complex patterns to a little bit of speech like that. And the model, you can see, does a very good job of reproducing these patterns for speech, but also for lots of other complex sounds. And in terms of variance explained, it captures something like 90% of the explainable variance across a wide set of sounds. So it's quite good. And it's good not only for normal hearing, but also for hearing loss. So you can see the difference between normal hearing and hearing loss is kind of obvious. There's just a lot of missing activity in the case of hearing loss. But there's also quite a lot more subtlety to the differences in the patterns that goes beyond just missing activity, and ICNet is quite good at capturing all of that. So then we have now these ICNets that we can use as encoder models, and we can stick them into this closed loop framework, and then we can train the hearing aid so that it restores the output of the impaired ICNet as close to the output of the normal ICNet in response to the original sound as possible. So I just want to give you a feel for this gap that we need to close. This is, again, another heat map to a bit of speech. And this speech is a lowish frequency vowel followed by a highish frequency consonant, so something like ash or something like that. And you can see that for the vowel, there's a little bit of activity missing. I should say that the units on the y-axis are organized tonotopically. So the units that are most sensitive to high frequencies are at the top of the heat map, the units sensitive to low frequencies at the bottom. So you can see the hearing loss in this case is a high frequency hearing loss, which is quite typical. There's a bit of activity missing from this low frequency vowel, and then quite a lot that is missing from the high frequency consonant. We can look at what happens when we use a typical traditional standard hearing aid to amplify these sounds before we present them to our gerbils. And what we see then is some restoration of the activity, but you can see for the consonant in particular, there's actually very little restoration, and a lot of a gap is left between what we would hope to see and what we actually see. These are just two different forms of signal processing that are used in current hearing aids. But when we look at what AidNet does, we can see that the restoration is quite a bit better. So it doesn't necessarily get perfectly back to normal, but it does get quite close under a lot of circumstances. And I've got the white arrow there pointing out what AidNet is doing that current hearing aids can't do is actually taking advantage of complex cross-frequency interactions in the cochlea. So current hearing aids will say, okay, you've got this much hearing loss in response to 4 kilohertz, so we're going to provide a certain amount of gain to compensate for that. But that's not really the way the cochlea works. The cochlea works by actually mixing information, across frequencies, and that can be taken advantage of by a hearing aid that has the flexibility to do that and can learn in a data-driven way the right way to trade off different frequencies against each other in order to get the neural activity pattern that it wants downstream. And that's the kind of flexibility that using a neural network allows you to take advantage of, whereas classical engineering doesn't. Okay, so to wrap up then, we are able to take this data-driven neurophysiological approach to hearing aid design. We do this first by training encoding models that seem to be quite accurate, and then we use those in a closed loop. And we are able, through this, to discover strategies that seem much more effective at restoring neural activity than current hearing aids. So what we're doing at the moment is trying to actually get these things to be suitable for actual real-world devices. And that means first tackling some issues of sound quality. When you optimize these networks on a purely neural metric, they don't necessarily come out with sounds that are as pleasant to listen to as you might want them to be. And then also figure out how we're actually going to optimize the parameters of the neural network when we don't have access to the intracranial recordings that we have in the animals. But this is stuff we're working on, and we're pretty optimistic we can get there. Thank you.
I'll ask a question, but if there's another question up here.
Go ahead, you want to change it over?
Hi. I assume that each person have their own individual auditory distortion. How would you adapt the network to an individual?
Well, I'll give a short answer as I can, but the way we train the models now is with a kind of shared encoder that would be appropriate for any individual, plus a conditioning module that would take some parameters related to the individual's hearing loss and reshape the sound processing to be optimized for them. So then it's only the parameters of that shared conditioning module that need to be fit for an individual, and the hope is that that can be done just with a bit of behavioral feedback in this very low-dimensional parameter.
I see. But, okay, you wouldn't be able to measure for that individual what would be the distortion. No. It would just be based on their reaction. Thank you.
Great, let's thank our speaker again. And next we have Alex Williams from NYU telling us about mechanistic interpretability of neurally predictive models.
All right, thanks so much for the opportunity to speak. I think it's a great question and topic of conversation, and I also really appreciate the breakdown into sort of the engineering versus scientific focus of the talk. So I think if I understood my assignment correctly, I'm on the kind of scientific sickos side of the debate of we just want to use these neurally plausible models to understand the brain. Oh, and I realize my slides are not up yet. Do I press something for that? No, good. Yeah, so great. Okay, so yeah, I'm on the scientific side of the debate of trying to use these models to make inroads in understanding a mechanistic interpretation of the brain, which is, of course, a really hard task. So probably my title was a little bit too ambitious, and I'd like to soften it a little bit and tell you that I'm just taking some humble steps towards this ultimate goal. Okay, so I'm going to start the talk by taking issue a little bit with how the question was phrased to us, which was, so you have a highly predictive model of neural data. Now what do you do with it? I think a more accurate and more frightening question would be, so you have hundreds, if not thousands, of really highly predictive models of the brain and neural data. What are you going to do with all of these models? If you look through the literature, there's lots and lots of evidence of this. Just to give one highly cited example, there's this paper from Colin Conwell and colleagues, where when you look at the details of that paper, they talk about doing a curated search over different models with various architectures, 224 diverse models, and show that all of them have very, very similarly high brain predictivity scores, despite having clear variation in their underlying representations. This is the main figure that's in that paper. The y-axis here are showing 2 different measures of neural predictivity, and the x-axis is all the different models that they looked at on a vision task. And you can see across both convolutional neural networks as well as vision transformers, you get very, very high predictivity with a relatively small spread across models. So remember this figure because I'm going to bring it back in a few slides to make a point. Okay, so we have many, many models to search over, and what this raises several questions in my mind. The first is, how are we going to navigate this really complex space of different models? And maybe for certain tasks, all models are fine and they're interchangeable, but maybe for other tasks, they're not interchangeable. How would we know? And a related question is, can we figure out ways in which these models are similar to each other, making similar predictions or having similar internal representations? And are there other ways in which those models are different from each other? A slightly more ambitious and future-looking goal would be, can we identify the kind of common structural features within these models that actually give us insight into what I'll call the mechanism? And that's a little bit of a squishy question, so maybe a more concrete goal to focus on is, can you strip away a lot of the extra complexity and parameters in these models and construct a very simple minimal model that is able to predict complex neural responses while still being human and interpretable? So these are the questions that I kind of had in mind when invited to give this talk, and probably I'm only going to give a very partial answer to a subset of these questions in the talk, and maybe some of these questions can also come up in the conversation. Work that my lab does, as well as many people, other people in this community, has really focused on the first couple of questions, and there I think we're making a lot of positive inroads. So how can we visualize and navigate the space of neurally predictive models? There's a lot of work that's been done on this, and it's not something that I can fully summarize within 8 minutes. So if what I say interests you for the rest of the talk, I would encourage you to take a look at this 3-hour tutorial that is on YouTube from the Cosine conference that I prepared earlier this year. I think it's a really good introduction and gives a good entry point into this space of quantitative methods. What I can try to do is summarize this entire 3-hour talk in a single slide. We'll see how that goes. You start by measuring neural activity across many different animal subjects, or equivalently train many models, neurally predictive models, and collect their hidden layer activations into these high-dimensional matrices. The key step is that we're going to compress all of those measurements into a distance matrix. So if K is the number of networks or the number of models that we're considering, we'll summarize that in a K by K distance matrix. And the cool result is that you can take this distance matrix and pass it to many off-the-shelf open source algorithms for regression, clustering, and low-dimensional visualization. In order to make that handoff really rigorous, it turns out that you want to define this distance matrix in a rather specific way. I'm not going to belabor the point too much, but you want these distances to be symmetric and have a self-consistency relationship with each other known as the triangle inequality. Those rules are not too hard to satisfy. There's many different metrics that satisfy those rules. And again, I'm not going to go into the details of this, but just to say that there's many different ways to do this, including many methods that kind of have its history and roots within this community. And again, for the overview, you can see the tutorial. What I want to spend my time on today is giving you one specific example of how you can use this sort of analysis, and I'm going to do it in a way that is not one of the traditional metrics that you may have seen before. So in particular, we're going to use neural prediction as a way to measure differences between models directly. So the idea is I have some dependent variable, such as human fMRI data. I'll call this variable Z that I'm trying to predict. And I have high-dimensional responses in 2 artificial neural networks, network I and network J. What I'm going to do is train a linear model to predict Z from Xi and train a different linear model to predict Z from Xj. And then I'll measure the distance between my models by computing the distance between those predictions on a trial-by-trial basis. So that is a symmetric notion of distance that satisfies the triangle inequality, and it's specific to the idea of comparing models based on their predictions of human data. So you can do this, and you can compute the pairwise distances across all networks that were considered by Conwell et al. This is what my postdoc, Sarah Harvey, did in this analysis. And you get this really rich representation of all the similarities and differences across those networks. And the point that I want to make on this slide is that this representation of the data gives you a deeper and more rich understanding of what's going on than if you just rank order the models from which is gives the highest R squared on neural data to the worst. To make this point, put even a finer point on this, you can take that k by k distance matrix across all models and ask what is a low dimensional embedding that is consistent with those distances. So that's what's being shown here. We're using a method called multidimensional scaling to reduce the dimensionality. And now each point in this space represents a different model, and 2 points are close together to the extent that those 2 models make similar predictions about the neural data that interests us. And we've colored the models by their architecture, and you can see immediately that there is some sort of clustering between the networks of different architecture. And again, this is maybe not completely invisible, but more difficult to see on the left-hand side in the way that people maybe traditionally plot these sorts of things. Okay, so summarizing and going back to the questions I started with, how can we visualize and navigate this space of many possible neurally predictive models? I would argue that we are developing a pretty mature toolkit for this, and I gave you a flavor of that. You can, again, look at the cosine tutorial for more details. Just because 2 models have similar neural predictivity scores, I think it's important to realize that their internal representations might be different, but also they may be making different predictions on a trial-by-trial basis, and that's exactly what we showed in this example analysis that I showed you. I don't think that this result is particularly surprising because neural data is generally noisy and sparse. So it's not like neural predictivity is a very strong constraint, and I imagine that there's many degenerate model solutions, and that's why we're seeing this diversity across models. Very quickly, to think about things more forward, future-looking, I would love to see, both in the work in my lab as well as the rest of the field, think about whether we can take a more mechanistic interpretability look into these questions. And this is something that neuroscientists have been very successful at in the past, and I think that we can carry forward this kind of perspective in modern times. I want to give a quick shout out to just one paper that I wasn't involved with at all that was led by Ben Cowley, which I think takes this approach of showing you can take a really complicated, large, deep network model that is neurally predictive and distilling it down into a much smaller system that might be actually interpretable. So we're very inspired by this work, and I have an intern this summer working along similar lines. This is very preliminary results, but I hope to come back next year and tell you more about it. There's also a lot of other work that I think this field could borrow from the adjacent fields in mechanistic interpretability and AI. But all of that work in mechanistic interpretability, or a lot of the work in mechanistic interpretability, I still have the issue of it usually takes individual networks and comes up with sort of nice just-so stories about that particular network. So I think the core problem that I'm kind of bringing to the fore in this talk remains that we have to grapple with the fact that there's many, many neurally predictive models out there, and we need ways to scale up this sort of understanding and analysis. And that's all I've got. Thanks.
Great, so we have time for one question.
Hi, great talk. I was curious what your thoughts are on, in a way you can functionally align human brains using like some kind of hyper-alignment algorithm. So I'm also wondering, when we are like measuring the model-to-model similarity, it's possible that the hidden units representing kind of similar kind of concepts might just be in totally somewhat different locations. So yeah, what are your thoughts on that? And yeah, if there could be, some kind of alignment that would get rid of those nuances.
Yeah, and I really apologize. I mean, this is part of the thing that I totally glossed over is how do you come up with these distance metrics? But a lot of the standard ones are using, like RSA style or an explicit alignment. So yes, like I think your question's directly on point, and we can chat more about the specifics about how you do that. But that is the rough intuition for how you make those measurements of distance.
Cool. Thank you.
Yeah.
Let's thank our speaker again. And next we have Ilis Roon from University of Amsterdam telling us about leveraging encoding models for insight in human scene perception.
Right. I also want to thank the organizers for putting this together. I think I'm also in the scientific application camp, but I really want to bring it back to CogNeuro and talk about how we can use these types of predictive models to address classic sort of questions in cognitive neuroscience. So as already explained, a central aim in cognitive neuroscience is to understand where and when visual information is processed in the brain. And our traditional paradigms, they use distinct conditions to identify spatial regions with fMRI, for example, and temporal profiles for specific types of stimuli with EEG, such as faces, objects, and scenes. And these isolated stimuli, however, they don't reflect our real-world visual experience. For example, in this picture of my kids in front of my house, scenes and objects and face information are all present at the same time. And for a really long time, the field really struggled with how can we study now the visual processing of such complex and naturalistic stimuli? What is happening to these spatial and temporal signatures if all this information is being processed at the same time? Now, as the organizers already explained, nowadays we have DNN-based encoding models that allow us to extract features from such complex images and then use these to predict brain responses both in space and time. However, standard encoding models typically flatten the representations across the image dimensions, so across the different, all the feature maps in a layer, for example. And this makes it quite challenging, actually, to quantify in a sort of really simple, intuitive way, the way we like to do experiments in CogNeuro, in which which image features are really driving this encoding performance. Now, of course, you can go mechanistic interpretability and try and distill features and so on. Here I want to introduce something maybe a bit more simple, where we use image-grounded encoding models to explicitly disambiguate the performance-driving features in separate models by fitting them to the same data, so essentially forms of model comparison. And the key idea, again, is not very complex. Basically, after fitting your standard main encoding model, you can go back and you can actually start to manipulate the inputs, or you can start to select or transform specific features in these models to see how that then affects the encoding performance. So I'm going to walk you through a bunch of examples of this. So this is all work done by Niklas Müller, a PhD student in my lab, who collected a large-scale EEG data set of big, high-resolution naturalistic street photographs from Amsterdam, which we call the Open Amsterdam dataset. And in a standard sort of DNN-based encoding experiment, you would use these scenes to predict the neural recordings by extracting the features for the image and then fit your encoding model based on that. But you can also train more models on the same data. So what Niklas did here is he additionally fitted models in which parts of the scene were systematically removed. So either the objects or the scene background. And then he extracted the feature specifics to the object or the scene background and looked at their performance alongside the full model. So here you see basically the performance, encoding performance on the test set for these different types of models. And as you can see, the full model is highly predictive. It's basically nearing the noise ceiling in the data. And of course, these reduced models where you black out parts of the scene are less good. It's lower than for the orange and the green is lower than for the blue. But the interesting thing now is that if you contrast these two reduced models directly, you can see that the dynamics are different. So they peak at different times, and scene information seems to be preceding object information. And this is really consistent with decades-old ideas that there's coarse-to-fine processing happening in scene perception. And here we now demonstrate that this is happening directly within an intact natural image. So we don't need to separately show backgrounds and objects. We're just showing that this is happening in these intact natural images. And we can take it a step further, and we can use this to study the representation of individual objects or scene elements in these images. So what happens to the EEG encoding performance if we remove one type of object from the object encoding model, or if we add objects to the scene encoding model? And we find some surprising things. So for most objects, the encoding performance changes proportionally with the number of pixels they have in the input. So there are, of course, many cars in these street scenes. And then if you remove these from the model, then the model performance drops quite a bit. But there's one exception, which are the trees. So removal from the object model, in fact, does not affect the object model encoding at all. And if you add the objects to the scene model, usually it doesn't really change that, but adding trees to the scene model actually increases its encoding performance. So this tells us that to the brain, or at least to the EEG data recorded from the brain, the trees seem to be part of the scene and not seen as an object. Now, for real scene perception aficionados in the audience, we can take this again a step further. Now we can use automated segmentation tools to identify pixels for different non-object-based scene elements. And we can see how they are encoded again by fitting separate models for each of these. And here we again see that some scene elements are more prominently encoded compared to their pixel level contributions. So it's higher for trees and vegetation again compared to sky and pavement. And we can do a lot more fun things with these encoding models. So we can do tests of classic sort of Biedermann-style scene schemas by shuffling the scene patches before feeding the images to the encoding model. And then we show that it strongly impacts the early scene encoding and shifts the peak to the later object encoding part. And similarly for objects, we can test how specific the encoding profile is to seeing these intact objects by shifting the bounding boxes and quantifying how much it reduces performance. And I just want to reiterate again that all these models are fed to the exact same EEG data. So we can do all these model comparisons on the exact same data from naturalistic scenes and then try to see really which parts of it really matter actually for the encoding performance. Now, here's another important factor of visual processing that we can address. So as I hope many of you know, classic CNNs, they sample the visual input quite differently from the brain, so they sample the input image uniformly, while the retina differentially samples center and periphery, and it amplifies foveal information through cortical magnification. So how much of that is actually showing up in the EEG data? So in a recent published work, Niklas used the same large-scale EEG data set that I just discussed to see how introducing a more differential spatial sampling, including how that basically affects the EEG response. So he included a retinal-inspired sampling transform, informed by the ganglion cell distribution in the retina, to the feature maps and compared how that affected the EEG encoding. And he found that introducing this transform at the feature level really improves EEG encoding, especially at the electrodes of the occipital, where V1 is, so that makes sense. But in fact, it improved encoding throughout the scalp, and it also allows us to quantify for each individual electrode how much foveal and peripheral information is actually reflected in the activity at those electrodes. Now, here's another example of how we use encoding models to provide direct tests of visual feature processing in EEG. So in this study, we collected EEG for original intact natural images at the top row, along with texturized and object-only versions. And then we quantified encoding performance by looking how well the features from the images themselves could predict the EEG responses. But we also looked at how well you could predict, for example, the response to an original image from the texturized image. And this is to address classic debates about the role of textures and object shape in perception. So here's what we find. So for textures, encoding performance is best when we use texture features. That makes sense. Participants view textures, features do best. Participants view objects, the object features do best, but now it gets interesting. If you look at original natural images, the texture features are in fact sufficient to explain the EEG response almost all the way to up to 200 milliseconds, or at least not sufficient, but they're equal to the original features. And only after that, the object features start to actually do a unique contribution. So that tells us that the early stages of natural scene perception actually appear to be somewhat dominated by texture information. Now, the final example, we can even apply image-grounded encoding models to more classic sort of cognitive neuroscience paradigms that are not using natural images, such as this one that aims to study how attention alters visual processing by presenting overlapping stimuli or isolated stimuli and asking how top-down attention is able to disambiguate these. So we fit separate encoding models to the EEG recordings for the different conditions, and we again cross-reference them across these conditions. So in this case, we try using 3 different encoding models to predict the response to this overlapping stimulus. And initially, the true stimulus which is the green one, provides the strongest encoding. But at about 250 milliseconds, the stimulus that was attended, so in this case the body, is just as good as the original one. So it's basically the red is approaching the green. And the non-attended stimulus, in this case the car, is much lower. So this really shows that the unattended stimulus is sort of suppressed and the attended stimulus is enhanced in the neural signal. And it pinpoints at what point in time this is happening. And now the main advantage again of the encoding model is that you can model this attentional enhancement and suppression in various ways. So you can fit an attention weight at the stimulus level or at the feature level, or you can maybe compare it to a pure neural encoding model, and then test how strong this amplification needs to be to accurately capture the data. And in this case, we find that an alpha, so an amplification weight on the features of about 30% fits the best, and that really nicely matches sort of existing literature on visual attention. And we find that at the individual level, this fitted alpha is predictive of individual differences in reaction time, so really how efficient the participants are at suppressing the unattended stimulus. So I will wrap up now by saying that image-grounded encoding models are powerful tools to study naturalistic image perception and vision in classic EEG experiments. And I showed you we can do hypothesis testing of the processing of distinct visual scene elements, such as objects or background, degree of spatial transformation, cortical magnification, necessity or sufficiency of specific image features, such as textures versus object features, and precise quantification of modulation by top-down or cognitive factors. I think we were asked to provide a hot take. I'm not sure if everybody got that note, but the one that I prepared was that, that I think it's not all about just optimizing performance. You can do lots of really creative stuff with these models by just playing around with them. And I think there are already a lot of examples of that at this conference. Just in the previous talk session, there were many of these, so I'm not saying that nobody's doing this, but I think we can do even more. And with that, I want to thank you and the people in the lab, and specifically Nicolas Müller, who did most of the work.
Time for one question.
Thanks so much for the great talk. So one hope with these encoding models is that you can go beyond the training data. Otherwise, we could just look at the original responses and be done. Mm-hmm. So when I'm looking at that plot with the size of the pixels of the objects that you've taken out, to me, that's basically the bigger it is, the more pixels you've blacked out, the further you are away from the training data. There's like, apart from the trees, there's like not much differential effect of like which object it is. So I mean, this is not specific to yours. I think like generally like there's the larger worry that like you go out of distribution, do they generalize in the same way that the brain does, and you hope that you've identified the whole function space?
So you're saying that you're moving because we're training the models on the masked images, so we're moving the input data, the training data away from the test data, and therefore the drop is due to the distribution shift? That's what you're saying?
Yeah, I guess ultimately, like if you had presented those masked images to the brain, then we would know, is the brain doing the same thing as the models on that, shifted distribution? But that's the thing we want to avoid, because
yeah, it's almost in a way that we're leveraging that, where we're basically saying, okay, how much do we, if you shift the distribution this way, how much is it reducing the encoding performance? And then we use that as a proxy for understanding how much that information matters to the brain. Yeah, I had something else, but I can't remember.
And let's thank our speaker again. And next is Thomas Nasalaris from University of Minnesota, telling us about concept dose responses in human visual cortex.
Thanks. I'm also on Team Basic Science. I'm going to talk about work by Mario Serafero in my lab, who's developed, I think, an interesting technique for addressing this problem. How can we map visual concepts at scale? So I'll start by unpacking what I mean by saying what our working definition of a concept is. For us, a concept, we're thinking about visual concepts specifically, and it's an aspect of the visual environment that's identifiable, so you know it when you see it. It's specific, so you know when you're not looking at it. And maybe most importantly, it's compressible into some kind of language, natural or visual language, so you can name it or describe it to others. Now, what do I mean by a concept map? Well, we have a lot of established examples, or at least some number of established examples of concept maps. So this is data from the natural scenes data set. We have digital flat maps of the cortex, left and right hemispheres of the cortex. We've labeled and bounded various visual areas that are been functionally identified in other experiments. And what I'm mapping here with color is a contrast between the activation from seeing faces as compared to a handful of other objects. And what we see is punctate activation and a set of regions that were originally identified and described by Nancy Canwisher and her colleagues, known appropriately as the face area. And this is just, we can do this for places and bodies, and what we're just doing here is showing a replication of some results that have been with us for quite a long time. Now, there's probably a handful of other maps, established maps I could show, but you know, it's maybe in the tens. I don't know, you discuss what you think your count is. And that's a far cry from the thousands of concepts that we humans are capable of recognizing and have interest in. So this is the problem of scale. There are lots of important concepts that we don't know how to map. I've talked to many of you, and I know that many of you are kind of just kept up at night by the lack of an established map for the dog jowl concept. This is a very critical, I think, gap in our understanding. I know that there's another group of you who are kind of in tension, who think dog jowl mapping is a waste of time and we should be really focusing on the bottom bun of hamburgers. And these two camps are kind of going at each other, but from my perspective, these are just two examples of the lack of scale. So to all of you in both camps, I'm here to help you with these, I think, interesting analyses. So we're going to start with an encoding model. In our case, it's the foundation model we're using is Dino V2, layer 12. We're going to try to predict activity in the natural scenes data set, and this map of the predictive accuracy measured by Pearson correlation of our model just shows you what you've seen before. We have yet another highly predictive or highly accurate encoding model for bold activity in this case. And all it really tells us is that there is some set of concepts that this foundation model has identified up to a dense linear map, which is this branch right here, that's encoded in the brain activity. But we don't know what those concepts are or how the activity is distributed. So in order to get at that, Mario subjected this foundation model, Dino, to the SAE treatment. So a sparse autoencoder takes the activity in foundation space, it expands it into a set of sparse units that are optimized to minimize loss, so to be able to reconstruct the original activity. Why would you want to do this exercise? Well, it surfaces concepts, and you can look at them. So if you take one of these nodes and you map it across, you map its activation across the stimulus by this round trip, you get a map that localizes the concept of interest. In this case, maybe it was a dinosaur. But there are a lot of, well, I mean, I would say I was just really surprised when Mario started showing me some of the concepts that were surfaced. After you trained up in SAE, I mean, we have bird concepts, so you've got maps of bird faces and bird chests. There are vehicle concepts, you know, the windows of trains, the engines of tractors. And many of you will be excited to know that he has surfaced a bottom bun of the hamburger concept, and even your precious dog jowl concept is there. So I think, you know, we have just a whole host of concepts, and if you're interested, you want to geek out on this, they have Mario and his collaborator Marlon Lee have a lovely Hugging Face tool that you can look at SAE analyses of lots of different foundation models. So how do we get back to the brain? That was all just AI side stuff. Well, we can use our encoding model to map concepts into brain space. So we can take one of these nodes and we pass it through the encoding model and we get a map of activity. Now, this is not a prediction of activity in response to a stimulus. It's some kind of brain space representation of a concept. In this case, it's a body concept. And as you can see, by the bounding of the extra striate body area that we're getting activation, or let's say concentration, where you'd expect it, and you can look at faces and places and get similar, familiar-looking maps. But this doesn't mean that we can trust this new technique for mapping novel concepts, the concepts you're all interested in. So how can we develop some trust in these maps? And I'm going to focus, or Mario's developed a technique that I think really emphasizes internal consistency of lots of a kind of a whole ecosystem of models. So what we're going to introduce is this idea of a concept prototype. So we're going to, again, start with our concept, in this case, zebra. We're going to use our encoding model to map it into brain space, and then we're going to use a decoding model to map it back to pixels. And what we expect to see is a concept prototype that looks something like the concept that we started with. So I don't know. I mean, this kind of mutant irradiated zebra, I think, is a good prototypical representation of zebra. But you don't have to just trust your eyes. We can quantify this by taking the image through a round trip. So we pass it back through the SAE and map the activation, and we see that it picks out the zebra. Okay? And we can do this for lots and lots of different, thousands of concepts. Each row here is an example of a different concept. Here is the vertical stabilizer of a plane concept. This is the reconstruction using Dynadiff and a comparison to the maximally exciting images just for consistency. We can quantify this too. Each row here is a concept prototype, and across the columns we're showing the SAE units that it activates. And if our prototypes are strongly and selectively activating the concepts, our intended concepts, we should see a nice bright line along the diagonal, which is what we see. I was concerned at first that these pretty pictures were restricted to just individual points, kind of maybe overtrained points in the foundation or brain space. So Mario developed another, I think, really interesting analysis, a steering analysis, to explore the spaces in between. So if we start with a stimulus activation map, we can, in brain space, we can push it in the direction of a concept, just additively. This lambda parameter determines whether it's a nudge or a shove in that direction. And as we increase lambda, we get a map of activity evoked by an image that comes closer and closer to expressing an intended concept. Across many concepts, we can get these nice, what we call dose-response curves. Along the x-axis, we're taking this stimulus map here and pushing it in the direction of the concept map, and we get this nice, graceful reactivation of the concept. What does this end up looking like? I think it's really pretty cool. What I'm showing you here, this is a reconstruction of a brain map in response to a surfer. This is the concept prototype that we ended up with for the sunset concept. And when you add them together in brain space, you get a surfer at sunset. And these are two different reconstructions using slightly different decoding models. If you add the cat concept to a tower, you get a cat tower. If you add iPhone concept to a man, you get a man holding an iPhone. If you add, I don't know, old-fashioned concept to a skier, you get a skier in the past. So this suggests to me that these concepts are not brittle, they blend gracefully. So collectively, I think this analysis, at least for me, it gives some trust. I feel like we can begin to think about using this in silico modeling environment or set of tools to map arbitrary concepts and to scale up the mapping process so that we don't have to have some poor grad student do the dog jowl experiment and the hamburger bun experiment. We can just generate them digitally and go to work trying to understand the logic. I think it's a real open problem. Like, no one, I don't think, would be able to predict what the concept map for stuff underwater would look like. This is a concept that arises. How is it mapped? I have no idea. If you scroll through the thousands of maps that have been surfaced by this analysis, I mean, you see all kinds of things. Montages, cityscapes of old, a lot of whimsical and mundane concepts. They all have their own map, and the logic of them, I think, is currently an open question. So I hope to next time, next conference, maybe we'll have something to say about that. As for my hot takes, we're doing interpretive work. That means you're going to bring your own opinion about what is important. I mean, some of you are obsessed with dog jowls. That's fine. I mean, that's fine. We can give you that and many other thousands of concepts besides. Something I've become convinced of is like superposition is real. I think we need decomposition methods. It's been really hard to apply it directly to brain activity, so we may be stuck with this in silico approach. You may have noticed that this dose-response idea involves a steering analysis. I think we need to be careful with that. I think it's potentially sketchy. I think there are really good important uses for it, for the development of prosthesis, for example. But I think there's some bad applications that make me wary. And finally, I don't know if generating thousands of these in silico maps is really going to help us understand the logic of concept representation, but I think it's a good start. We'll see how it goes. So I want to end by thanking my lab. Again, shout out to Mario. This is mostly his work, and his collaborator, Marlon. They have a poster tomorrow if you want to learn more about the details. Thanks.
Oh, thank you for the great talk. You showed that the SAA features can predict activity in specific brain regions. Could this framework also be used in reverse direction, for example, to optimize or generate images that selectively evoke a target region?
Yeah, you think to search for like maximally exciting stimuli for the brain?
Yeah.
Yeah. I mean, I think it's a tool that could be used. They could be used for that potentially. It's another application for sure. Yeah.
Thank you.
All right, and our last speaker is Hannes Mehrer from EPFL, telling us about moving toward ANN brain models for the clinic.
Hi, thanks so much for the organizers. I really enjoy talking here today, and I'll go right in. This is the usual setup that we use in many like encoding model studies, where we feed the input images to our subjects. So we present them to the subjects, we record the neural activity or the behavior so that we get the data in one specific format, and we use show the images to the same to the models and extract the activations from there in order to get the data in the same format. And then we can relate these matrices, so like what the model predicted and what our subjects showed, in some way, be it linear predictivity, RSA, or some other form of alignment metric. And I want to highlight two studies that have been using this framework and have been doing model-guided sensory stimulation to drive neural responses. The first one is from Bashevan and Carr from 2019. That's their science paper, where they use a vision model, an intermediate layer of the vision model that they linearly connect to the V4 neurons in a monkey, and then use this model in order to synthesize stimuli that drive these neurons. And in order to, from the machine learning perspective, do model evaluation, they then show these stimuli, these synthesized stimuli, to the monkeys again. And what we get is the following, where on the x-axis we see the model predictions, and on the y-axis we see the actual responses from the V4 neurons in monkeys. So the blue dots are responses to naturalistic images, whereas the red dots are the synthetic images, where you can see not only the model predicts higher responses than to naturalistic images, but also in the V4 neurons, there we can see that this is quite a bit higher than what you see to naturalistic images. The other study that I want to mention in this context is the one that Greta Tushkute already presented herself, so I'm going to go over this quickly. So they used a language model approach to select sentences to drive or suppress the language system in the human brain, and then checked again whether for subjects in the scanner that see or like that are presented with these stimuli, whether that really drives what the language selective cortex. And this is indeed the case. Like the drive sentences do drive so the language selective cortex, and the suppressed stimuli also do their job in the intended direction. And these studies and many others in the field, for good reasons, use subjects that are in good health. What I want to look at is basically a little bit of a diversion from that, and that is look at dysfunction. Or to put this in one sentence, we want to build stimulus computable models of the brain and behavior in a disordered or dysfunctional state. The first example that I want to give here is revolving around the idea of visual prosthesis. So here we have model-guided neural stimulation that steers visual behavior. There's quite a bit of work that has been done in this space without any sort of modeling, for example, by Peter Rosema's lab from Amsterdam, where they use UTA arrays in monkey early visual cortex, where they have retinotopic organization, in order to steer the activity patterns that they apply to the cortex to induce simple, like the percepts of simple objects, such as letters. And what we'd like to do, we'd like to extend this in order to induce more complex visual objects. And to do that, we will move to a higher level cortex, specifically IT. And since there's no retinotopic organization that could help us to determine what kind of pattern should be applied in order to induce these percepts, we are helping ourselves with topographic vision models. So these are vision models with a notion, or like an internal notion of space. And this can help us to simulate the first neural and then behavioral effects of causal intervention techniques, so here, microstimulation. What we do in this project are three main steps. The first one is we do a model brain mapping. And here, what we want to do is basically say, if there is the UTA array in the monkey, which is obviously fixed, how do I place the in silico UTA array on the in silico cortex such that it best matches what the functional responses in the UTA array in the monkey look like? Once this is done, we can run the experiment that we want to do in the monkey first in the model. And in this way, we can select those combinations of the stimulus and the stimulation site that yield the largest behavioral responses. And once this is selected, we basically hand over the stimuli and the stimulation sites to our experimental partners in Amsterdam. So this is work that the experimental part was done by Paolo Papale in Peter Rosema's lab. And they then run the stimuli in order to see whether we get the same output. Before I get to the results, I just want to shortly explain what experimental design has been used for this. And that was a simple delayed match to sample task where the monkey is trained to tell us which of the two alternatives is perceptually closer to the first stimulus. And this then gives us two psychometric curves, one for the trials where there were stimulation and one where there were no stimulation. So this is pink versus black. And the behavioral effect that we're measuring is the difference between these curves. So a large behavioral effect would be the pink curve is shifted to the left, whereas the black curve stays where it is. Now the big question is, how do we model this in a model that is only feed forward and doesn't even have like a notion of time? What we've done here as a simplification is we feed these three images of interest to the model, extract the activation from the last layer, and then compare the last layer activations pairwise. So always the stimulus with one alternative, and then check whether the stimulus versus the alternative is the higher similarity than versus the other alternative. And if so, this is counted as a saccade to the left, vice versa, a saccade to the right. And in this way, we can get from the model the same psychometric curves that we would get from the monkey. So this then gives us the model predictions here on the x-axis and the actual monkey responses on the y-axis. And we can see that these are highly correlated for the two monkeys together, but even if you do this analysis separately for each monkey, the correlation stays about the same, they're both significant. From here, one last sentence to that. So I think this correlation is far from perfect, but it's in my eyes, first step towards using ANN brain models to guide causal intervention techniques that could help us with some translational question, for example, building a next generation of visual prosthesis. The next thing I want to present is model-guided font optimization for individuals with dyslexia, and that's a project headed by Melika Honermand. Here, the neural basis of dyslexia is that the visual word from area is reduced in its activity in dyslexics versus controls. And on a behavioral level, there's a reading impairment, whereas other cognitive skills are not impaired, such as visual IQ. Now the question is, can we find visual word form selective units in the model, ablate them, and then get some behavioral readout? And before I give you the results, I'm just showing you what we did in order to ablate the units. So in human neuroimaging, we would identify the visual word form area by a classic functional contrast of words versus other objects. And we did the same with the model in order to identify those units that are most selective to text. And from here, we can either ablate the visual word form selective units or, as a baseline, the same number of randomly selective units. And as a last step, we have a behavioral readout. This could be, these are basically the tests that are administered for dyslexia screening in schools or hospitals. So this would be some specific dyslexia screening test here called ROAR, some other from Luke et al. And on the visual IQ and reasoning side, we use Raven and Kempler. And then what we see, if we ablate the visual word from selective units, we get the model to become dyslexic per the clinical definition, as in it's below a dyslexia threshold, whereas Raven and Kempler, the visual IQ and visual reasoning tests are unaffected. But if we do the ablation in the random units, we see not quite dyslexia, but we see that in Raven and Kempler, so in visual IQ, there are also changes happening, so not so much like matching what we'd see in dyslexic patients. So long story short, what we see then here, if we ablate the visual word form selective units, we see a reading impairment, but no visual IQ effect. And this then allows us to use this model to do further experiments with this model. So this really shortly before I wrap up, what Melika has been done is using the model in order to identify which font is best for this dyslexia-induced model, in order to then see which one can we use the model in order to develop a font that works even better for, or is even dyslexia more friendly for dyslexic people. So what we see here is that for fonts that are recommended for dyslexic individuals, they work better in the dyslexia-induced model, whereas those that are difficult for people with dyslexia, they're also difficult for the model. To wrap up, I want to say we've seen there are some approaches first approaches of using model-guided microstimulation to steer visual behavior. We can use this in the context of dyslexia, and Melika has also done a follow-up project where she used the same approach, but in a different context, so like for a different disorder, specifically to induce anhedonia, one of the two main symptoms of depression, to have that in the model, and then similar to what she's done in dyslexia, as a basis to develop either diagnostic or therapeutic tools. And that's it from my side. Thank you.
Great, we have time for a question, and we'd also like to ask our speakers to return to the stage for the panel.
Hey, thanks for the wonderful talk. My question is, how would you tackle individual differences, either in dyslexia or depression, using the model approach? Can you think of a way?
Yeah, it's a good question. I think the only claim that we have so far for the models that we've done this with is that we induce this on a group level, so not so much on an individual level. That might be feasible as well, but this is something that we're currently working on.
Okay, with that, let's give one last round of applause for our speaker. Okay, so for the last 20 minutes of our session, we want to, I guess I'll stand way over here, all the seats are way over here. Okay, So we're going to have a little bit of a panel discussion. And to start this off, I just want to sort of remind everyone of a quote that I think that we're all very familiar with, which is, all models are wrong, but some are useful. And I think that part of the motivation for having this session is that oftentimes it seems like we focus on addressing the very first half of that, saying that all models are wrong, how do we make them better? But really, I think that all of these talks today have highlighted ways that these models actually can be useful. Some of these encoding models can be useful. So that said, I actually just want to ask the panelists to raise their hand if they think that neural encoding models that we have today are good enough in terms of things like prediction accuracy to be useful for enabling additional scientific insight or translational or engineering impact. So raise your hands if you think they're useful. Okay, great. What about the audience? Can we raise hands? So useful and then not useful? Okay, I love this. I think that if we would have asked this 5 years ago, it would have been a very different response. Okay, so with that in mind, what I actually want to think about a little bit more is for each of you what it means to have a model that is useful. So for a particular question or approach that you are interested in, so for instance what you talked about today, what test or tests would you subject a model to in order to convince you that it is good enough for that purpose?
I could go. Yeah. I think I would just take the pragmatic explained variance view, even if you have a model that explains just a tiny bit more variance than your predictors before. You know, for language cortex, right, it's a hard problem to figure out like what are the features and properties represented, and we have some interpretable features we can use, and then we have like simpler models like word co-occurrence models, and if you add an LM and it just adds 5% of explained variance, that's a win, right? And you understood that there's something about those representations that help, right? So I think as soon as you just get past like whatever you had before, like I'm pretty happy then.
I guess I'll add just a little bit of a wrinkle on top of that. Like explained variance is fine for certain questions. I think there was a really nice question from somebody in the audience about like generalization performance, like explained variance on what the networks were trained on or what are natural images that you train your encoding stitching model on. And I think there's a bit of an open question of how well do these things actually generalize. Like even for things like finding maximally exciting images, it seems like this is empirically useful. But I mean, if you look closely at the plot that was shown in the slide, it is falling below the unity line so the model is predicting that these exciting images should be producing even larger neural responses than they actually are, which is a sign that they're kind of extrapolating beyond their training data. So I think that's like an interesting wrinkle that I don't know. Maybe there is like really good work out there that characterizes that, but that's an important point.
Yeah, I agree. I think that generalizability is the new benchmark, the new goalpost to achieve, and there are more and more stringent forms of generalizability. I mean, early encoding models, I remember it being somewhat novel importing this idea of cross-validation from machine learning and just testing the prediction accuracy of a model on a held-out data set as opposed to just like, I don't know, doing a significance test on the model weights. And that I think is like baked into our process now, and that's great. But now I think, at least in the neuroimaging field, what a lot of groups are thinking about is, well, if I have an encoding model for person A, do I need to collect another 40 hours of data on person B to be able to use that? And we need to get to a point where we have, you know, the Rosetta Stone encoding model, you know, maybe constructed on someone who's subjected themselves to 1,000 hours of scanning, I don't know. And that's all we need. And there's some fine tuning with a minimal tractable amount of data. I think that's when encoding models are going to get really useful, particularly for engineering applications. There was a question about the auditory prosthesis, the hearing aid. You know, if we know how to do this kind of generalization across subjects, we're going to be in good shape.
I think I want to answer this question in the context of like translational research or research that goes in the direction of like clinical research. There specifically, if we think about like neurological and psychiatric disorders, if you were, as we're trying to do, and I think some others as well, try to induce these disorders in the models, then I think the gold standard in the clinic is really that some clinical staff administers a clinical test, and then basically like the model passes that test in the sense that like it shows like pathological behavior. But you can do this only, let's say, in silico, as in like we've set up like clinical tests and like run them like with a Python script. But you could also do some like basically like further version of this where like you set up a web interface and then have an actual clinician interact with the model and then confirm that this is like pathological in the sense of the clinical definition.
I just wanted to add one follow-up to what Thomas said about the generalization. I just want to give a quick shout out to the revision initiative. So in the introduction, you guys put up the Lion fMRI dataset, which is a newish 70 large-scale image fMRI dataset, and there's an ongoing initiative to use that dataset to try and replicate many of the findings that we have so far on, for example, datasets like NSD, but also the dataset has baked in several specific generalization tests, which have to do with the overlap, the distribution shifts between the test set and the training set. And I think this is, if you're saying, you know, the next step is to think more deeply about generalization, I think this is a good setting to work with that. And we're basically inviting the whole community to participate in these, in trying to see what replicates, what generalizes. That said, another answer to your question. So I think for some things, like in the examples that I gave, it's not always the most important thing to have the most best predictive model. Like if, for example, for the object and scene comparisons, it's not, I don't think the results would be very different if we would use Dino features instead of the, maybe it's even AlexNet features that we were using, or ResNet features. Like it's about the within model comparisons that we're doing. It's not about the specific, the most predictive features that, for the particular thing we were interested in, right? So it really depends on what your question is.
And so that actually, I think, leads nicely into kind of the next question here, which I guess I was wondering if there were any perspectives of what, so you've outlined a lot of things about what makes maybe how we can test our models in order to maybe make them useful. But are there differences between what would make a model more useful for a translational purpose versus more useful if we're really interested in using it for some sort of scientific discovery?
To me, I think enhancement of our models that would address both translational and basic science concerns, I think is along the lines that Hannes was showing, is if you have a model that you can perturb, you can ablate it, yeah, but I mean, if you can make it depressed or get it high, or give it, a psychiatric condition, and then make it predict the brain's, activity in the brains of people who are in the same state of mind, I think that's going to be really powerful. I think we're pretty far away. We need to add more biological detail. We probably need to add noise to our models, dynamics. They should probably have some kind of spontaneous activity. None of the models we discussed had any of that. When they do, then maybe we can start using them to explore various states of various different kinds, qualitatively different kinds of minds. And I think that will be very powerful, both for science and translation.
Nick, did you have something?
Yeah, I think related to that, I think you can get pretty far with engineering or translational applications, even if your model is not biophysically plausible. I think that plausibility maybe helps for regularizing or providing useful inductive biases or something, but you can get pretty far without it in certain contexts. I think if you want to develop better scientific understanding of how the brain is doing what it's doing, then the biophysical plausibility is kind of non-negotiable, right? So I think that's one clear difference.
So concerning the clinical research or models that we try to make, be it depressed or represent some other disorder, I think for many, not all, but many psychiatric disorders, it would be a very interesting target to do basically pilots for pharmacological research in these models, but you would either need like one of two things or both, and that is like either the model represents like molecular changes that are coming from like administering some drug, or you would have a way to show on a systems level what the molecular changes are actually inducing on the systems level, so that you could use the same models that you use currently. And I think that is completely unexplored.
So I guess I want to move into a slightly different question, which is like sort of moving forward, right? So moving forward, I think that Iris kind of said that we want to continue to push forward and start to really take these models that we have, which seem to be really doing interesting things, and use them for science or use them for engineering purposes. So with that in mind, and some of this has been mentioned a little bit, but is there infrastructure, benchmarks, data sets, maybe even cultural norms that we really think that the field should adopt in order to better utilize our encoding models and move them towards scientific or engineering progress? So basically, is there something that you can think of that's really holding us back right now that we should start to do in the coming years?
I think, as Iris pointed out, there's been so many actually really awesome new projects for replication and using encoding models, sharing them online. I think that's really been really cool to see. I think from a language perspective and also related to other higher level cognitive abilities, it's data is still a huge limitation, which I'm super interested in. You need the right kinds of data to do these kind of comparisons, right? You can't just take any data set that was collected 20 years ago with the same analyses in mind that we're trying to do these days. And so I'll definitely say that in my domain and some related domains, data definitely remains the largest bottleneck.
And while maybe another panelist answers, we will open the floor up for questions. So if anyone has questions, if you could come up to the microphones here.
This might venture a little bit into hot take territory, but I'm not sure that we're actually very good at benchmarking our models in a way that makes sense for science. I mean, generalizability is actually one thing that I wish we had better benchmarks for, so it's great to hear that people are thinking along those lines. I also think, getting back to one of the points I was making in my talk about trying to strip away complexity and find simple models that might even hurt in lose a little bit of performance, but nonetheless explain 95% of the explainable variance. I'd like to see, some sort of subsidy that you would get on a leaderboard for maybe a model that is not top performing, but is actually much simpler. And yeah, I guess people might disagree with me on this, but when I look at a lot of benchmarks that people do, not even necessarily things like brain score and stuff, but just NeurIPS papers, benchmarking different models, it feels to me like the really simple, linear models are not particularly well tuned, and usually you just benchmark several different deep nets against each other and pick whichever one has 1% better performance. But the simpler baselines are not given the love that they deserve.
So I guess one of the things that was brought up before was out of distribution data sets in particular. And so how do we encourage the field to collect that sort of data? There's many different ways that you can make your data out of distribution. And so I guess I'm curious, do you have thoughts on what type of data do we need? Do we need behavioral data? Do we need neural recordings? Do we need fMRI? Do we need EFAs? Anything along those lines?
Yeah, just quickly, I think, there's a lot of simple things you can do, like increase contrast, very, very high contrast natural images. I think that's something that's shown that can really move hidden layer activations in deep nets very far. And again, this is something that maybe this does exist and I've just missed it, but biological data that looks at these sort of weird, quasi-naturalistic but out of distribution images, and it would be I would be interested to know if you train a deep net on actual natural images, does it generalize and predict how the brain responds to these kind of quasi-naturalistic manipulations? I'm not aware of too much work along those lines.
Okay, so we'll start over here.
Okay, actually, I was going to ask a different question, but just the types of out of distribution stimuli should just be experimental stimuli in laboratory studies in psychology. I mean, there's just,
Sure, yeah.
so, I mean, the simplest thing, which is not often discussed, is we got lots of classic experiments for 100 years now into cognitive psychology and perception. Those are out of distribution that test very particular hypotheses. Like,
Totally agree. There's quite a lot of things in the space of interesting out of distribution.
So, and we're putting together a data set that has those stimuli called in mindset. But the question, you raised the question about, you know, what's the difference between developing a model for scientific versus scientific things? And I think one thing I imagine is, you know, building at scale bigger and bigger data sets of images, for instance, is probably, or auditory stimuli or whatnot, is no doubt excellent for engineering purposes, makes models better. But I don't think that scaling these things up and up larger and larger is good for scientific purposes, because one of the challenges is to try to count the data efficiency in human development. So if you just train a model on, you know, 2 billion images and get a better brain score, I mean, you're doing it, but you're doing it in a qualitatively different way. So, I mean, there's a nice contrast in my mind between, you know, engineers, you don't care, bigger data sets. But if you want to develop a scientific model, you got to be constrained in language. You know, one of the tricks is children learn quickly. In vision, children learn vision quickly. And so if you just build bigger and bigger models with bigger and bigger data sets, you may well be developing models that are just using qualitatively different ways. I mean, that's what you need to do, develop a model that works with limited data.
I would argue that if you're an efficient experimenter, there's no distinction there at all, actually. Because I think even if you're interested in big data and big performance, like, and you want to collect like 2 billion images, yes, you can go and sample them from whatever we like go and look at every single day, but you're not going to span the space very efficiently, and that's ultimately also going to limit your performance in your downstream model. And that's exactly where, like, if you know about all these manipulations you could do to images or sentences from like psychology, like neuroscience, then you just build those in, and if you do 2 billion with those manipulations, you like double win. That's like, so in my head there's really no distinction if you run these experiments.
But the trick here is that, sorry, no, the last thing, I can stop.
Yeah. I think we should go to another question just for the sake of time.
Similar point about, I think engineering progress is much faster than the scientific progress. So something I always found inspiring is how David Cicillo started using these models, like back in the days, the RNNs on the data from Krishna Shenoy's lab, training them on the system, and then afterwards trying to reverse engineering them, taking them as a goalpost of like, this is how far I get with a linear model, this is how far my deep learning model get. Can we understand what the different mechanisms, can it teach us something new about the system? I know one study that did this in V1, basically starting with a Gabor filter bank and a CNN and trying to bridge that difference, which turned out to be divisive normalization, which, you know, we might have known in advance. But what I'm trying to get at is like, how far are these models from helping us to reverse engineer V2, let alone V4? At what point are they going to suggest to us the mechanism that will allow us to hook that up?
I'm not sure if they will be able to like tell us on a mechanistic level like how V2 or V4 or so works, but I think like what these models can do, like the current models that we have, is that we use them for like exploration of like, let's say, like selectivity clusters across the cortex. And this might not be like the, let's say, scientific insight in the sense of like mechanistic insight, but it might tell you something which you can then like more easily explore like experimentally, because yeah, the exploration is cheap in the model, but it's expensive with human subjects.
I think that we'll take one more question and then wrap up because they will kick us out of the room.
I feel like I need to defend the honor of my department to say we definitely should have known about divisive normalization going into that study.
Hi, so I was wondering what are your thoughts on when we're thinking of computationally modeling different kinds of neurodivergence. I think the individual differences in what one matters a lot, and I think that traditionally the way the individual subject encoding works, it's a lot like, okay, we have the representation from some ANNs, and then we try to linearly predict one's brain, and maybe we have a separate encoding weights for a different subject, but But that still kind of more or less assume that like these subjects could be linearly predictable from each other in a way. But when we are going far into have like some practical applications for neurodivergent people, I think that that might just totally not work. So if not linear, then what kind of assumptions one would use? And if you think that linear assumptions might still work.
I hope so. I mean, I'll just tell you my ideal. I don't know if it'll work, but it's kind of related to what I was saying earlier. I mean, there's some shared component of variance that's shared across all of our brains. And if we had one model that captured this like ultra just basic generic brain, then everyone, you know, the stuff that makes each one of us individually interesting would be the residual between our brains and that kind of platonic model. So that's a program I think that's not obviously going to fail, and it could work. And depending on how similar our brains are, you may not need a ton of additional data from every individual to figure out what makes them interesting, whether they're neurodivergent or whether they have, you know, just some other interesting characteristic that you want to reveal or understand. That's a hope. I think the same modeling framework will hopefully apply broadly.
Yeah, I think there's no reason the individualization needs to be linear. You might want it to be in the end for some reason, but you can start with something more flexible than that just to prove that you can do it, and then work backwards from there. Once you've got something that works, you can try to strip it down and make it simpler, but I don't see why you would need to start it with it being linear in the first place.
Okay, any 30-second hot takes to end on, or should we end?
Well, I would encourage everybody who's building models for the purposes of understanding to try to put themselves on the hook with some applied context to judge whether they actually have improved their understanding when they've built the better model or not. I think we have been, you know, quote unquote, understanding how hearing works for half a century, a whole century, and yet the best we could do to come up with a hearing aid based on that understanding is to build a simple amplification device. I think that's rather telling about how deep our understanding of hearing really was, and only when you put yourself on the hook can you, really be forced to come to terms with that.
Let's disagree. Okay, I think putting yourself on the engineering hook has turned out to be weirdly unrevealing, at least in the case of vision. We have a lot of really powerful vision models that, as far as I can tell, don't use a ton of the basic cognitive neuroscience or the basic neuroscience. I mean, it's inspired by it, but it doesn't use a ton of the basic neuroscience that we've learned, and yet they perform really well. So I think it's under constraining to have an engineering success. The problem of how our human brains do vision remains a problem.
Okay, with that, let's give a final round of applause for our panelists.